Let's talk about tRNA. What are tRNAs used for? Well, they bring amino acids to the ribosome during translation and match them up with the codons of the mRNA. tRNAs are about 75 to 90 nucleotides long and form the secondary cloverleaf structure, which you can see here. The 3' end is called the aminoacyl end, or the acceptor stem, since this is where amino acids bind to. This sequence is CCA in the 5' to 3' direction, or ACC in the 3' to 5' direction. And it's really important to be careful in how these directions are denoted, because when we talk about codons, we talk about them in the 5' to 3' direction, and that's why this is referred to the CCA codon instead of the ACC codon. So the 5' end would be here, and the 3' end is here. You can use this mnemonic to help you remember, and it stands for kin-carrier amino acids, since this is where amino acids do attach. The opposite end contains an anticodon, and this area is complementary to a codon in the mRNA that it will bind to. It's this matchup that allows each codon to correspond to an amino acid. The T-arm, which you see over here, contains a T-Cy-C sequence, which stands for thymidine, pseudouridine, and cytidine. And this part is necessary for tRNA ribosome binding, so you can think of the T-arm as the T-arm tethers tRNA molecule to the ribosome. The D-arm, which you see over here, contains the dihydrouridine residues necessary for tRNA recognition by the correct aminoacyl tRNA synthetase. Think T-arm detects tRNA by aminoacyl tRNA synthetase enzyme. They are less important to know compared to the acceptor stem, but may be necessary to be aware of for your exam. You may get a question on step 1 that basically just asks you what type of molecule might contain pseudouridine or dihydrouridine, and your answer choices may be something like DNA, mRNA, tRNA, microRNA, etc., and you'll know that it's tRNA that contains these molecules. Before it can do its part in translation, a tRNA must first be charged by adding its corresponding amino acid to it. And here is the amino acid that binds to the acceptor stem. This is an ATP-dependent process catalyzed by an aminoacyl tRNA synthase enzyme. And there's a different enzyme for each tRNA. So here we have a tRNA that corresponds to methionine, so this might end up at the start codon. And the aminoacyl tRNA synthase for methionine will first check if this is indeed a methionine tRNA, and if it is, it will attach a methionine amino acid to the 3' end. If tRNA is mischarged, it still reads the usual codon, but inserts the wrong amino acid. For example, in this case, if tRNA was mischarged, it would read the codon as AUG, but insert another amino acid, not methionine. So this step requires 1 ATP, just as we mentioned. And once a tRNA is charged with its amino acid, a protein called initiation factor 2, or IF2, is needed to get it into the ribosome so it can find its codon. And this is where we have the codon-anticodon pairing, which you see the process down here. Since this is a vital process, we can use antibiotics such as tetracyclines that target the 30S subunit of the bacterial ribosome, and this can prevent attachment of the aminoacyl tRNA and essentially kill the bacteria. I talked about tRNA wobble in another video, but I'll briefly go through it again. Basically, the idea here is there are only 20 amino acids, but there are 64 possible ways to combine 4 different amino acids in a sequence of 3. So each amino acid tends to correspond to multiple different codons. For example, proline is coded for by all of these codons, which differ only in their third base. So if there's a mutation in that third base, you'll still end up with the same amino acid in the protein. Therefore, this third base doesn't matter as much as the other two, and this area is called the wobble position. Now, this isn't always the case. There are some amino acids that are only encoded by one codon, but by and large, the ones that are coded by multiple codons only vary in this third position. Now, a quick quiz. What sequence does the acceptor stem code for? That's CCA, and you can think of that as can carry amino acids.